All right. Well, let me talk about some of my current and former projects. So one of the projects I'm working on has to do with renal cancer. And in fact, actually, it's clear cell renal cancer. I had a guy, he's a doctor out in Pennsylvania. He gave me a call. He saw my videos on YouTube. And he said, hey, I like what you're doing with bioinformatics. I watched your video where you kind of show how you don't even need your own data. I work on renal cancer, and there's just absolutely no really good markers for the advancement of this disease, number one. Number two, potential therapies that you might use on this cancer. He's like, I've seen what you did. Can you actually mine the public data and go get a bunch of renal cancer data and tell me what I should be looking at? I was like, sure, I could do that. And just like with any cancer, there's stages. Obviously, stage 1 to stage 4, and by stage 4, it's invaded its tissue, and then it's spreading to other organs. There's different types of kidney cancer, and as you can imagine, they all have their different survival outcomes, and that's why we really care about classifying kidneys or different types of cancer. Is if we classify it, will it have any effect on that patient and the therapy that we do to him? You can see by far the most abundant cancer here is clear cell. About 75% of anybody that has renal cancer has clear cell. So I decided to focus on this. And as we were talking, and as I was getting involved in this project, he said, hey, listen, there's hardly a lot of variability there's not very much variability as far as the therapies go and how we treat this. And in fact, most of the therapy is basically targeting the angiogenesis or the bloodstream to the tumors. And these work with variable results. And then they also use mTOR inhibitors. So basically, they have all these different types of renal cancers, but they basically only have 3 potential types of cures for this cancer. So I said, okay, why not? I can do this. So basically what I did is I used this is a program called Correlation Engine. And again, this is plugged into basically all the public data that's available that's worth looking at. So what you can do is you can actually search this by a disease. So I just put in clear cell carcinoma of kidney. When I do that, 113 studies come up, right? And I think people that do research, they kind of think, I always want to tell them you don't have to reinvent the wheel. That people have been doing research in your area for a long, long time. There's not very many of us whose research is so specialized that we can't find something out there that's related, right? So I found these 113 studies. So what I wanted to do was, in these 113, I looked for studies that had comparisons of the different stages of the cancer. So when I did that, what I did was I compared, say, grade 1 to normal tissue, grade 2 to grade 1, grade 3 to grade 1, grade 4 to grade 1, and then I had a list of just studies where they looked at metastasis versus the primary tumor. And again, so in this particular study here, say grade 1 versus normal, I found 7 studies looking at that, and basically what I did was I only took those genes that were differentially expressed in 4 out of those 7. And you can do that for all these. So basically, if you kind of add up all the research that I'm using here, I mean, it's millions and millions of dollars worth of research to get potentially an answer for him. And so when you do that and you do it like a meta-analysis, so all the stages are here on this line. These boxes are the different stages, and they go from less severe to more severe. If the gene went up, it's significantly in that study, it's red. If it went down, it's green. And what you can tell, and when I do these meta-analysis, you can just see all these genes just pop up, right? And for the most part, they're very consistent. Some of them actually go up right at the beginning when you're looking at normal tissue, and then you kind of lose that protein as it comes. And then you'll see some like this that are kind of variable, up in some and down in others. This is a really interesting one. Anyway, this is probably characterizing the different carcinomas in here. But mean, this is how you tell this worked, is that there is no way aldehyde dehydrogenase 6 family member A1 isn't somehow involved in renal cancer if it's differentially expressed in all but one of our stages, right? So not only do we have just kind of the basic data, but now I can take this data and cross-reference it with clinical data. So there's another data set. So this basically mines the TCGA data set. It's called Cohort Engine. And so what you can do is you can actually look up these genes and basically pull out survivor curves based on what they have in their database. And what you can see here is with this gene, when you have, high expression in the green, your chances of survival are much better if you have low expression of this gene, right? And I had to find this one. What's interesting, it's so cool. Once I jump into like these projects, I learned so much about all these different types of cancers. And what you find is most cancers, you would think you would have all these oncogenes, but it's mostly what the cell is doing, just shutting down the things that are controlling those oncogenes. So most genes go down with cancer rather than go up. But you do find some like this TNF-SF14, this transcription factor, or no, I think it is. Anyway, when you have high expression or low expression, you do better, but when you have high expression of this thing, your chances of survival are much worse. So again, what I'm doing is I'm using all this data out there to provide them with targets. You can look for biogroups that are overrepresented, and I think that's one of the key things about this is that I have all these genes associated with renal cancer. Are there any particular biogroups that pop up? And obviously, a lot of blood vessel stuff. And so it makes sense that these people that they're trying to inhibit VEGF, these blood vessel stimulating kind of hormones, they're using as a therapy because a lot of the genes that are associated with renal cancer are associated with blood cell development. And again, we can get all the genes that are associated with that category. This is the really cool part, is that now that I have a list, so basically what Illumina has done is also taken a bunch of chemicals. They have information on chemicals. And so basically, they'll have studies where people use a chemical and then it changes gene expression. And so what you can do is based on the genes that you have, you can determine whether any chemicals are there any chemicals out there that might have a positive or negative effect on your genes of interest. So that's what I did as I took the grade 4 versus grade 1. I took all these genes and I looked for something that might have a negative correlation so that if it goes up. So if my gene goes up in my study, in this study would go down, and vice versa. So when you actually look at that, the top one here is this, I can't even say that, naphthalene-diimide, blah, blah, blah. Again, no one's heard of this as a potential therapy for breast cancer. I work on way too many diseases. Renal cancer. But if you look here, I started looking it up, and like, what does this thing do? And sure enough, they're starting to use this as an anti-tumor agent, but they're only using it in pancreatic cancer. So again, for him, I was like, hey, now you have another chemical you could potentially use in your clinic, or at least try it out maybe on some cells first, obviously. That's a bioinformatics. Just throw it on some people. He's like, I can't do that. All right. Should. All right. So, and again, this is kind of typical of what I would do, is that somebody would come to me with a problem. Usually, sometimes they have their own data, but it's so bad that I usually talk them into like, okay, I know your data's here, it's really nice and everything, but look at all this stuff out there. And usually what I'll do is I'll mine basically all the public data that's out there based on their project and basically kind of come up with these solutions. Here are the genes that I think are important. Here's why I think they're important, because they have clinical implications. Here's a drug you might use on your experiment to actually, help manipulate the system.